Hello, Biology 100 students. Welcome to your lecture on the nervous system. Or actually, I'm going to call it the best of the nervous system because usually in a human anatomy and physiology class, we would spend 4 or 5 lectures, probably the last quarter of the semester, talking about the central nervous system, peripheral nervous system, special senses, all that. But we're going to do most of that in one lecture. So keep in mind, this is not a comprehensive lecture. It's just sort of the best of or greatest hits of the nervous system. Okay, so what are the functions of the nervous system? If we boil them down to 3 main functions, we see that it's sensory, integrative, and motor. First of all, we'll start with sensory. So sensory is bringing information into the central nervous system, such as information from our eyes, information from our touch receptors, and then that information is processed in the brain and spinal cord where we analyze that information, we interpret it, and that is called integration. And finally, given that information, we might make a decision to do something. We might want to move our arm or something like that, and that is called motor output. So motor output involves nerves that travel down to our skeletal muscles, but also to our glands and cause our muscles to contract or activate the glands. All right, so this diagram just shows the 3 processes we were talking about in the last slide. So firstly, we have sensory input coming in there. You see there's a glass of water on the desk. So that information travels via sensory neurons from our eyeball, and that travels to the brain, where the brain does that integration. It does interpretation. It says, yes, that is a glass. That glass looks like it has water in it. And the brain also says, I'm thirsty. I'm a little bit dehydrated. And so it might generate a motor output. That is, it's going to send information through a motor neuron, which we'll talk about, to a skeletal muscle. So you go and pick up that glass and have a drink of water. So a lot of times our motor output can be voluntary, for example, when you're picking up the glass, but sometimes it is involuntary. And that's called a reflex. Take a look at what's happening here. You can see on the left-hand side that somebody is swinging at a baseball and that bat has broken off. And you see that shard of the bat that's flying into the stands, and we see it right here. And this sports photographer just happened to get a really great shot of what's going on. So this shard of bat's flying into the stands, and you can see the people are all looking at it. They've got their hands up, like that. This guy right here is, and then this guy, I love, he's got food in his mouth right there. But all of them have their hands up, okay? And the hands up here is, of course, a protective function, right? Their brain is processing this information, saying, oh my gosh, this bat is coming towards me, hands up, right, to protect the face. The other thing you can see that's going on is the cringe reflex this guy has. So his neck muscles, his platysma is all stressed, and you can see the fright face going on, and that serves a dual function. It helps to alert others you might be talking to that something is going on that we need to pay attention to. So all this is happening very quickly, and it's a reflex because it's involuntary. We don't have to think about it voluntarily. Our central nervous system is making that decision for us. All right, to lead this lecture out, we're going to talk about the organization of the nervous system. So the nervous system is kind of organized into 2 main areas. We have the central nervous system, which includes the brain and spinal cord, and then we have the peripheral nervous system. This is everything outside the central nervous system. So we're talking about the nerves that are going down your arms like that, the sensory structures that are in there as well. And later on, we'll talk about the fact that there's a sensory division and a motor division to our nervous system. So the sensory division brings information in to the senses, or from the senses to the brain and spinal cord, and the motor division brings information out. And we have a voluntary motor division called the somatic motor division, and an involuntary division called the autonomic motor division. And we'll talk about this one towards the end of the lecture. Okay, just like any other body system we've talked about, the nervous system is based on lots of different tissues, right? We have nervous tissue, of course, but we also have blood and connective tissue and things like that. But the neurons are the most important cells, and these are the cells that generate action potentials or nerve impulses. And the purpose of these cells, the neurons, is communication. Communication between the senses and the brain, communication between the brain and the muscles. In addition to having these neurons, we have other cells called neuroglial cells. So neuroglial cells are helper cells. They don't generate nerve impulses, they don't generate electrical potentials, but they do help out in other ways. Okay, so let's start out first with looking at a neuron. So this is an idealized neuron. We see that it has a cell body up here at top. That's where the nucleus would be. That's where the majority of organelles would be. And then coming off that neuron, we might have dendrites. Now, dendrites are like little antennas on that neuron that receive information from adjacent neurons. Neurons are always talking to one another, and so here's where we get that information. There would be another neuron that's attached to that or very close to it, and we'll talk about synapses later on. Coming off that neuron is a long process called an axon. The axon is sort of like the electrical cable of the neuron. This is where we form the action potential right here at the axon hillock, and that electrical charge travels all the way down until we get to either another neuron, or in this case, we're going to a muscle cell. So the axon is going to be traveling quite some distance. Cell bodies of neurons are generally going to be in the central nervous system. So the cell bodies are in the brain or spinal cord and also in ganglia. But the axons themselves can travel outside the central nervous system to your muscles. For example, think about the muscles that run your fingertips there, right? These muscles right here, the flexor digitorum, their cell bodies of those muscles are actually located in your spinal cord, but they have the long axons that go all the way out to those muscles. And finally, I want to point out we have something called the myelin sheath that helps to insulate that axon, and it's made up of our neuroglial cells. And one of the things that helps us to do is to speed up nerve impulses. Okay, there's 3 different types of neurons in the nervous system, and basically they're paralleling the 3 functions that we've talked about so far. So we have sensory neurons, interneurons, and motor neurons. So sensory neurons bring information from the outside, from our senses, into the central nervous system. So here you can see we have some skin and maybe you've stepped on a nail or something like that. So that's going to be bringing information about pain, and that travels this way into the spinal cord. And within the spinal cord, that sensory neuron will synapse or connect to possibly another neuron. This is an interneuron. Interneurons do interpretation, identification. Remember, the spinal cord is not conscious, but we're still going to be analyzing and determining whether it's a good stimulus or a bad stimulus. And eventually, if we want to do something about that stimulus, we have a third neuron type called the motor neuron. And the motor neurons bring information out of the central nervous system and take it to our effectors. Now, effectors are things like muscle cells or glands or things like that. So this stimulus here is painful. That information is going to be processed inside of our spinal cord, and the spinal cord may make the decision to contract the muscles of the back of your leg and pull your foot off of that nail so it won't be so painful anymore. Okay, so we said that neurons are the cells that generate action potentials, that transmit the nerve impulses, but we do have these neuroglial cells that help out. And there's actually over 7 or 8 different types of neuroglial cells. There's only 2 that I want you to be familiar with today. And one of those are called the Schwann cells, which are up top. And the other ones here are called the oligodendrocytes. These are cells that help to insulate the axons. The Schwann cells insulate axons in the peripheral nervous system, and the oligodendrocytes myelinate cells within the central nervous system. So this myelination or insulation does a couple things. It helps to speed up nerve impulses so they travel much quicker. And the other thing it does within the peripheral nervous system anyway is this myelin sheath can enable us to repair an axon that becomes damaged or severed. Not so much in the central nervous system though. Okay, so this is what the myelin sheath would look like within the peripheral nervous system. You can see that we have an axon here, and that's that big cable coming off a neuron, and that's where a nerve impulse travels. And around that we have 2 big Schwann cells here. And so the Schwann cells insulate parts of that axon, and as a result, the charge will jump between the individual cells. This is called the nodes of Ranvier, and the nodes of Ranvier are basically just gaps in the myelin sheath that allow depolarization or charge to jump from node to node. So big picture here is by having this insulation, we're really able to speed up nerve impulse transmission a whole lot. Okay, while we're talking about myelination, we should talk about gray and white matter. You've heard about this before if you've watched any movies about like JFK and stuff like that, and they're talking about brain matter and it's getting really grisly. But here we're talking about gray and white matter within the central nervous system. So gray matter here refers to areas that are darker because they have lots of cell bodies. White matter is referring to areas that don't have cell bodies, and it's primarily myelinated and non-myelinated axons. So if you look within the brain, for example, you can see that all the gray matter is up towards the surface, the cortex right there, and then white matter is on the inside there. What I want you to think about when you think about gray matter is that's where the connections are. That's where the processing units are. That's where all the important electronics of your brain are located right here, just under the surface of your skull, whereas all these structures in here are actually just myelinated axons. So this is the wiring that's taking information from the left to the right or up to down, but it's not really the processing units. The processing units here in the brain are right on the outside. On the other hand, if we take a look at the spinal cord over here, you can see that the gray matter is located inside the spinal cord. So that's where the cell bodies are. That's where the neurons are talking to one another. And then the white matter, the axons and such, is located on the outside. Again, here's just another picture of a brain, and we've taken, what is that, a superior to inferior section off there. And you can see a little bit of that gray matter on the very outside of the cortex. And again, that gray matter is where we have the cell bodies. The white matter is primarily where we just have axons, no cell bodies to speak of. Okay, now we're at the big part of the lecture. So here you need to pay attention. We're going to talk about how information is transmitted along a neuron. We've already said that it's transmitted as an electrical potential called an action potential, but now we're going to talk about how that action potential occurs. Okay, so there's three electrical phases we need to talk about in terms of our neuron. Firstly, we need to talk about the resting phase. During the resting phase, the inside of the axon is negatively charged, and we'll talk about why that is. Now, if we stimulate that neuron enough, eventually it's going to depolarize or form an action potential. During the action potential, we switch that internal charge from negative to positive. And finally, repolarization goes back to negative again. Okay, this slide just shows a diagram of the various potentials we might see inside of a neuron. So firstly, starting off on your left-hand side, we see something called the resting potential. Now, the resting potential for most neurons is around negative 70 millivolts. You don't really need to know the number, but you do need to know that it's negative, right? Negatively charged, and we'll talk about why. If we stimulate a neuron enough, we cause that charge to go up a little bit, and that's called a graded potential. And if we have enough of that graded potential, we eventually hit something called threshold, and then we have our depolarization. Now, the important thing is that not all neurons are going to reach threshold. It depends on how much stimulation is occurring and how frequently that's occurring. So not to be blunt here, but it's sort of like a sneeze or even an orgasm. If you need enough stimulation, eventually you get to threshold, and then bam, depolarization, repolarization, and then we have something called a refractory period. So during depolarization, we're going from that negative 70 up to a positive number like positive 40 here, and then repolarization takes us from positive 40 back down a little bit more negative, back to negative 70. So eventually we get back down to that resting phase here. So you should be able to tell me what the charge is inside of an axon during depolarization, repolarization, and resting potential. Okay, in the next few slides, we're going to concentrate on neuron physiology, and we're going to talk specifically about why the neuron is negative at some phases and positive at others. So please pay attention. So the drawing here just shows part of the neuron axon. Remember, the axon was that long tube-like process that propagates and generates the nerve impulse. Now, inside that axon, we have some proteins. The proteins help to contribute to the internal negative charge, along with some ions. Remember, ions are charged atoms. And so the two atoms we have here that are charged is we have a potassium ion and a sodium ion, and both carry a positive charge. How did they get to be positive? Well, they both gave up electrons. Okay, what you can see here, though, is we have something called the sodium-potassium pump. The sodium-potassium pump uses active transport. If you remember back to our lecture on osmosis and diffusion, we said active transport could push things against their concentration gradient. And that's exactly what's going on here. We're pushing all the sodium ions to the outside and the potassium to the inside. And this is done at a 3-to-2 ratio. 3 positively charged sodium are pushed out, and 2 positively charged potassium are pushed in. So we're creating a charge differential. There's 3 more positive things out here for every 2 positive things in here. So relative to one another, the outside of the axon is positive, but the inside of the axon is negative. And that's why we're in resting phase. So if you take a look up there in the top left-hand corner of your screen, you can see that we're at approximately 70 millivolts, and we're negative, and that's because most of the positively charged things are out here. Now, think about what would happen with simple diffusion. Where does sodium want to go? Well, sodium's more concentrated out here, less concentrated there. So just keep that in mind. Now, right now, sodium can't move because the sodium gate right here is closed. So sodium is just hanging out until we open the gate. So what causes the gate to open? Well, if we stimulate a neuron enough, we cause these gates to open, and that's exactly what happens. When the neuron becomes stimulated, the sodium gates open up and sodium rushes inside. Why did sodium rush inside? Because it was more concentrated out here than it was in here. Now, when sodium rushes inside, what does that do to the overall charge? Well, it becomes much more positive on the inside because now we have all the sodium or a lot of the sodium in there along with the potassium. So if you take a look at the top left-hand side again, you can see we're in the depolarization phase. We've gone from negative 70 millivolts up to some kind of positive 40, somewhere around there. So the reason the neuron becomes positive during depolarization is the sodium gates have opened and we've let all that positively charged sodium inside of our axon. Now, what's the next phase? Well, the next phase is something called repolarization. During repolarization, we're going to open up this other gate right here, and this gate only lets potassium to cross. So where is potassium more concentrated? Well, it's more concentrated inside the cell. So which way is potassium going to go by diffusion? Well, if you said out of the cell, you're exactly right. So when all that potassium rushes out, it causes the charge inside of the axon to become relatively more negative. And that puts us back down into what we call repolarization. So if you take a look at the left-hand side there where the arrow is, the charge inside the axon is going from positive back down to negative again as that potassium flows back out of the axon. And finally, we need to talk about restoral of the resting potential. Remember, the resting potential was definitely negative. Well, how do we end up getting sodium back to where it needs to be and potassium where it needs to be? Well, we simply let our sodium-potassium pump do its work while both of the gates are closed. So remember, the sodium-potassium pump was pumping out 3 positively charged sodium for every 2 positively charged potassium that it was pumping in. And because we were more positive on the outside, less positive on the inside, this puts us down at a charge of negative 70 millivolts. So we're now back at our resting membrane potential. Okay, now that we've talked about the physiology of nerve impulse formation and the resting potential inside of a neuron, we're going to talk about how neurons talk to one another. So we used electrical charges to talk across the neuron, but between neurons we usually use chemical signals called neurotransmitters. Okay, so this picture is just showing a close-up of 2 neurons connecting to one another. So here was an axon of the first neuron, we'll call that the upstream neuron, and here is a dendrite of the downstream neuron. And here you can see something called the axon bulb. Now the axon bulb is where we secrete our neurotransmitters. Initially, that first neuron was conducting information as an action potential, electrical charge, but when it gets to this end bulb here, what will happen is we will release something called neurotransmitters. And these are little vesicles of chemicals that travel across a very microscopic distance, and this area is called the synaptic cleft. So a synapse is where 2 neurons connect to one another, and there is a gap there called the synaptic cleft, and the neurotransmitter diffuses across the synaptic cleft, and then it binds to the second cell. Now, what that second cell does depends on whether that neurotransmitter is stimulatory or inhibitory. If it's stimulatory, it might make this second cell form its own action potential. But if it's inhibitory, it actually will make it less likely to form an action potential. All right, so normally I would spend a long time talking about the different types of neurotransmitters, excitatory, inhibitory, etc., but we don't have that time in this class. But I do want to say something about psychoactive drugs because it's something that most of us can relate to. Now, psychoactive drugs, whether we're talking about something that's a recreational drug that's illegal or something that's a legal drug, they basically modify the nervous system in a couple ways. One, they might simulate or act as a neurotransmitter. It might be some drug that acts as the neurotransmitter dopamine. The other way is it can block neurotransmitters from working, or the third way is it can keep neurotransmitters from being reabsorbed. Usually when we have neurotransmitters secreted by this cell and then going to the other cell, they only reside in that synaptic cleft for a short period of time. So after a while they're either broken down or a lot of times they're actually sucked back up by something called reuptake. And so if we're treating a psychological disorder, let's say somebody has depression, we can treat them with something called an SSRI, or selective serotonin reuptake inhibitor. Now, serotonin is a very common neurotransmitter and one that we think has to do with a feeling of well-being. And some people that are clinically depressed may have lower serotonin levels. And so when we use this SSRI, it actually prevents this serotonin from being sucked back up so quickly, so it keeps it in that cleft for longer and it stimulates that second cell for a greater period of time. And as a result, that second cell might fire off its own nerve impulse. So a lot of psychoactive drugs either simulate neurotransmitters, they inhibit neurotransmitters, or again, they might prevent them from being destroyed or sucked back up by their first neuron. Okay, now we're going to talk about the central nervous system. Remember we said the nervous system consists of the peripheral nervous system, which is all the peripheral nerves, and then we talk about the central nervous system, which includes the brain and the spinal cord. And we're going to concentrate mainly on the brain. So firstly, the spinal cord. What is it? What is it for? So the spinal cord is very important. I don't want to say that it's not, but we're going to spend one slide on it. So the spinal cord is, among other things, a conduction pathway for sensory and motor impulses. Remember, sensory information is coming into the body. And so imagine you're walking down the beach, you're looking at somebody, and you're not paying attention where you're going, and you step on a glass bottle or something like that. That sensory information will travel up through nerves in your foot, to the spinal cord, and through that spinal cord we'll conduct that information to the brain where we actually perceive pain and think about, oh, I want to take my foot off that broken beer bottle or whatever. So that's one thing it does. The other thing is it brings motor information from our brain back down to the muscles. So if you step on a broken bottle, you might want to pull your foot back off that broken bottle so you don't continue to damage your foot. And that would be an example of motor nerve impulses. The other thing the spinal cord does is it does integration. It actually does some decision making but we're not consciously aware of it. So if you do step on a broken beer bottle on the beach, you will eventually register the pain in your brain, but before you do, your spinal cord has probably already made a decision to pull your foot off of that sharp object so you don't continue to damage your foot. Again, I wish we had a lot more time to spend on the spinal cord, but sadly we don't. I do want to point out this is a spinal cord in a transverse section, and here you can see the cord itself, which is really about the diameter of your index finger in most places, and we can see spinal nerves coming out right here. Now, as you get older, like I am, you have discs that are sitting right here, cartilage discs that are separating the individual vertebrae. Eventually, those discs can rupture or herniate, and they impinge or kind of apply pressure to that nerve, and that can cause a lot of pain. So protect your back, because once those discs rupture or deteriorate, there's no repairing them without surgery, and it can be very, very painful. Okay, now we're going to talk about the brain, and the brain is sort of the superstar of the central nervous system. It does so many different things. Firstly, it helps to process information that's coming in from our senses. For example, if you see a person walking down the street, your brain helps to recognize whether they're male or female and helps to recognize who they are if you know them. The other thing the brain does is it stores information. It stores, for example, your social security number. It stores your credit card numbers if you happen to memorize those. It stores phone numbers, etc. It also oversees the running of automatic body functions. And we don't think about this very much, but your brain is monitoring your heart rate. It's monitoring your respiration rate. It's monitoring your blood pressure, even though you're not aware of that. Of course, the brain also helps us to make decisions and take actions. If you're driving down the road and all of a sudden brake lights go on in front of you and the car comes to a screeching halt, you're going to want to make a reaction too and probably put on your own brakes. So it helps us to do reflexive actions and also voluntary actions as well. And finally, the brain is the center for intellect, emotions, and behavior. All of that's located right here in the prefrontal cortex. Okay, this slide just gives an overview of the different parts of the brain we're going to talk about. They include the cerebrum, which you can see right here. They include the thalamus and hypothalamus, which you cannot see right here. And they also include the cerebellum, which is down here, as well as the brainstem. So we'll spend a little bit of time talking about what each of these different areas does. Okay, before we go on to talk about the functional areas of the brain, I just want to mention that there are some hollow parts in the brain called ventricles, and these ventricles are filled with something called cerebral spinal fluid, or CSF. Now, the function of CSF is to maintain an ideal osmotic and ionic gradient within the brain and its tissues so that we have optimal nerve impulses. The other thing it does is it helps to transport a little bit of waste products around there as well. Now, the CSF is generated up here in something called the choroid plexus, and it basically travels through all these different ventricles, and it also travels around the brain within the meninges. Eventually, the cerebral spinal fluid is sucked back up by something called a arachnoid villi up here, and that helps to put that fluid back in the bloodstream. Here you can see a cross-section through the brain showing the various ventricles right here. So we have the two major ventricles, left and right ventricles you can see here, that would be filled with fluid. Now, sometimes the cerebral spinal fluid mechanism goes haywire. The mechanism that's supposed to reabsorb the excess cerebral spinal fluid doesn't work, and we get something called hydrocephalus. So hydro means water, right? Cephalus here means head. So it's literally waterhead or water brain. And as a result of this, we have kids that are born with these huge heads up there because the cerebral spinal fluid is not being sucked back up and put back into the venous circulation. So we have to go in there and usually put a shunt in, and that shunt drains the cerebral spinal fluid, ironically, down into the abdominal cavity, and that's just fine. These kids go on for their whole lives, and they probably need it replaced 2 or 3 or 4 different times. But hydrocephalus is a very treatable condition if we get to it early. Okay, now let's go on to talk about the functional regions of the brain, and the first of these is the brain stem. So this is the brain stem back here, and it consists of the medulla oblongata and the pons, and these areas basically cover very basic life support functions. We're not consciously aware here, but it is a very important region because it controls our heart rate and respiratory rate, and that's here at our medulla oblongata, so right there. It controls heart rate and blood pressure, which keeps us adequately perfused, and it also is one of our respiratory centers. The pons, on the other hand, also aids in respiration, but it's also a bridge or gateway between the spinal cord and the cortex, the higher areas of the brain. So pons literally means bridge. Okay, the next functional region of the brain is something called the cerebellum. The cerebellum is located in back of the brainstem, so it's this orange region right here. And the cerebellum helps with a couple things. First and foremost, it helps to assist with muscular coordination. Anytime you make a voluntary muscular movement, it involves your cortex up here sending impulses to your muscles saying, I'm going to try to grab that remote. But what your cerebellum is doing is it's taking information through the sensory receptors in your arm and also visual information and making adjustments so you hit your target every time. So that's the function of the cerebellum, maintaining a coordinated muscle contraction. Now, when you are inebriated and have had too much alcohol to drink, the cerebellum is one of the first areas of the brain to become incapacitated. As a result, your coordination goes out the window. And this is the reason why we can use field sobriety checkpoints to see if somebody's inebriated, because if they are, their cerebellum's not working properly, they can't do the heel toe and so forth. The other thing the cerebellum is good for is it stores skilled muscle movements. So sorry if I can point right here. Look at this guy who's a professional pitcher in baseball. Chances are he's played baseball or practiced baseball for tens of thousands of hours during his lifetime. And so he can probably throw the same pitch each time again and again, and because the muscle movements involved in that pitch have basically become hardwired and stored within the cerebellum. Another example here is people that play the violin or really any musical instrument. If you played and become a master, you have gained proficiency in your storing those motor movements, those muscle movements in your cerebellum and also areas of the cortex so that you can play the same way every time. Okay, our next functional region of the brain is something called the thalamus. The thalamus is located right here above the brainstem. It doesn't look like much. It's a circular region, and it's called the interthalamic adhesion. And basically, it is a relay station. Information travels up the spinal cord, goes to the thalamus, and the thalamus routes that to the appropriate region of the cerebral cortex. Now, the thalamus also helps us to register a sort of crude recognition of pain and touch and things like that, but it doesn't help us to localize that to different areas of the body. That goes on up here in the cortex. And finally, we also think the thalamus plays an essential role in learning and cognition. Okay, our next brain region is something called the hypothalamus. Now, the word part hypo means below, so the hypothalamus right here is just below the thalamus. And even though the hypothalamus doesn't look like a very impressive region of the brain, it's very important. It has so many different body functions. In fact, if you ever get to an exam question and you don't know the answer, if one of the potential answers is hypothalamus, heck, go ahead and choose it because it's likely the right answer. The hypothalamus controls lots of things. For example, it helps to control our autonomic or automatic nervous system, which we'll talk about towards the end of the lecture. It also coordinates the endocrine system. Shoot, it actually runs the endocrine system. It's in control of the anterior and posterior pituitaries, and those are also control of lots of other endocrine glands. The hypothalamus also regulates things like our hunger drive, our thirst, our sex drive, and also controls body temperature and circadian rhythm. Okay, next is an area called the epithalamus. Now, epi means above or on top, so the epithalamus is located above the thalamus, and it's a very small area of the brain that's located back by the midbrain. There's two different parts, one called the pineal gland and the other one called the habenular nuclei. Now, the pineal gland is the gland that secretes melatonin. Melatonin is important in setting our circadian rhythms. We talked about those briefly on the last slide when we talked about the hypothalamus. Your circadian rhythms are when you wake up and when you feel sleepy, you want to go to bed. So that's set in part by our epithalamus or our pineal gland. The other part of the epithalamus is this habenular nuclei, if I could pronounce that, and that involves forming memories associated with smells, which is kind of weird. So think about what your grandma's house smells like or what your auntie's house smells like, right? They have particular smells, and if you smell that smell, it probably brings you back. Think about your first boyfriend or girlfriend. Did they wear a cologne or something, and you smell that cologne on the street 5, 10 years later? It really brings you back to that same memory because that memory is associated with particular smells, and that's part of our habenular nuclei, which is part of our epithalamus. Okay, so now we're moving on to the cerebrum, and the cerebrum is the higher brain region. It's the largest brain region in humans because this is where we do a lot of our reasoning and also processing stimuli, etc. So the cerebrum is separated into left and right hemispheres by this longitudinal fissure right here. And the hemispheres are able to talk to each other by a special structure called the Corpus Callosum, which unites the left and right hemispheres. So again, the cerebrum is the seat of intelligence. It's where we have our communication skills, our intelligence, our analytical ability, our memory, and also our ability to generate motor nerve impulses, as well as to process sensory information coming into the brain. Okay, if you remember back to the beginning of the lecture, we talked about gray matter and white matter, and we said that white matter was primarily myelinated axons. It's basically the cables that go between neuron cell bodies. On the other hand, gray matter is the actual neuron cell bodies in the areas where two neurons connect. So if we take a look at the cerebrum, the largest area of the brain, we can see the gray matter is located towards the surface and the white matter is internal to the brain. And this is important because what this means is all the important processing equipment is right on the surface of the brain. So even a mild concussion or something like that can greatly affect or impact the tissue that's up in here. The white matter, again, are just myelinated axons that's carrying information from one processing unit to another processing unit. So here we can see the distribution of other white matter within the brain. And again, these are called tracts. So tracts are just big clusters of myelinated and non-myelinated axons that are carrying information between different regions of the brain. Now, probably the largest and most important tract within the brain is something called the corpus callosum. The corpus callosum is a bundle of white matter that allows communication between the left and right cerebral hemispheres, between the left brain and the right brain. In fact, there's been some people in the past that had such severe seizures that the only recourse for treating these seizures was to go in and cut the corpus callosum, and sometimes that would actually alleviate the seizures, but it also resulted in some kind of peculiar abnormalities, sensory abnormalities and processing abnormalities. So if you want to hear more about what happens when we cut the corpus callosum, click on the link at left. Okay, so on to the cerebral cortex. The cerebral cortex is a very complex region of the brain, and it's divided into 6 lobes, 5 of which we can see here. Now, these lobes are named for the bones that lie over top of them in the skull. For example, the occipital lobe right here is directly underneath the occipital bone, and the occipital lobe is responsible for interpreting visual information. So visual information comes from the eyes way at the front of the brain, but that information is actually processed and analyzed back here in the occipital lobe. Now, the parietal lobe involves processing information from skin receptors and touch receptors, so identifying things of touch. So think about the last time you reached into your handbag or your book bag, you're feeling around in there, you're not looking in there, but you're trying to identify things by touch, and you find something, you're like, I'm not sure what that is. You're pretty good at figuring things out because you have this stereogenesis, the ability to form a three-dimensional image of something you're not actually looking at just by touch. So this is a pretty important area of the brain. Next, we have the temporal lobe. So the temporal lobe is located underneath the temporal bone, and it helps to analyze and process information that is auditory. For example, speech and music and things like that. And lastly, we have the frontal lobe. The frontal lobe is truly the most miraculous lobe of the cerebrum because this is where personality and intellect are located. So the frontal lobe includes the prefrontal cortex, which again is the area of intellect. It's the area of complex learning, which is cognition. It's the area where we have our working memory, where did I lay my keys? And it's also where our personality is located, as well as our judgment and moral character. So essentially everything that makes you is located up here in the prefrontal cortex. Now, we didn't always know what the functions of these various brain regions were. A lot of times we didn't know until somebody got an injury that affected that brain region and somehow affected their personality. The example that I want to give here is somebody called Phineas Gage. Now, Phineas Gage was a railroad foreman back in the 1800s, and he was working on helping to build a railroad. And part of that involved drilling big holes in a rock bed and then filling those holes with blasting powder and tamping it down and then blowing them up. Well, his job was to tamp down that powder with something called a tamping stick. And as he was doing that, he must have created a spark because the powder ignited, and this tamping rod, which is a 3-foot rod, you can see here, flew through the air, went through his head all the way clean through, and the people there thought he was dead. He went down immediately, and here you can see his skull. It's got this huge contusion in there, a huge hole in there. But he sat up within a few minutes and drove himself in his little horse and buggy to the local Sawbones, the doctor, and said, this should be work enough for you. And Phineas Gage actually lived for several years after that. He wasn't paralyzed or anything like that, but his personality changed vastly. Where before he was a God-fearing man and a hard worker, he became now very impetuous, and he also used profanity all the time. So this damage to his prefrontal cortex helped scientists to recognize that the prefrontal cortex is where we store information on judgment and personality. So while we're on the subject of the prefrontal cortex, I want to mention something called a lobotomy. Now, lobotomy was a surgical procedure that was performed in the early 1950s, 1960s, but is thankfully now in the past. And a lobotomy involved basically severing some of the tracts up here in the prefrontal lobe. Initially, they did burr holes in here, but there was a particular physician, Walter Freeman, that figured out he could do it simpler and quicker if he just stuck ice picks above the eyeballs and twirled them up there to sever some of the nervous connections up there. Now, why would you do that? Well, back at this time, we didn't really have a lot of psychoactive drugs we could use to treat mental illness, seizures, and things like that. So his option was, oh, let's just cut some stuff up there and see what happens. So again, initially it started with surgeries, but by the end he was doing these procedures in his doctor's office. He would simply take an electroshock therapy machine and shock the people into unconsciousness, and then stick these ice picks, these surgical tools, up above their eyeballs, go through the bone, crunch, and then sever those tracts. In this case, he was treating a 12-year-old boy who was just a little bit obstreperous, and he would basically talk back to his mother. So if you want to hear a story about what that little boy is like now, you can click on the link below. Okay, before we shift gears and move off of the brain, I want to talk about some diseases that affect the brain and central nervous system. The first of these is something called meningitis. A meningitis is an inflammation of the coverings of the brain called the meninges. Now, we didn't have time to talk about those, but the meninges do form a protective layer around the brain and also around the spinal cord. Now, meningitis might be caused by a viral pathogen or a bacterial pathogen. It's particularly bad if it's bacterial. It can lead to encephalitis and even death. So I remember years ago when my brother was in college, he was actually at a sorority or fraternity party where everybody had been drinking beer out of red cups and stuff like that. 2 days later, somebody that was at that party died in the hospital, and it turns out this person had bacterial meningitis, and it's very virulent, particularly among younger people. They had to go back and sort of contact trace everybody at the party and made sure they all got on a very powerful antibiotic so they also didn't come down with meningitis. Okay, the last disorder of the central nervous system we're going to talk about is something called a CVA, or cerebrovascular accident, otherwise known as a stroke. Now, strokes are the third leading cause of death in the U.S., and they occur when the blood to the brain is interrupted, causing death or ischemia up there in the brain tissue. Now, it's most often caused by a blood clot, and it can lead to permanent hemiplegia, where you can't move half your body, but prognosis is greatly improved when we recognize the symptoms early and treat them fast. And so memorize this right here. It says act fast. So we're looking for deformities up here in the face. Is one side of the face not symmetrical? Is one side sagging a little bit? The other thing is look at the arms. Have the patient come out and basically push on your hands. If they're giving uneven strength between the two different arms, it's an indication they might be having a stroke. Finally, speech is a really important indicator. If they're slurring their words and they're not, drinking or something like that, that can be an indicator of stroke. And of course, time here means we have to act fast. The faster we get that patient into the hospital and treated with anticoagulants, the quicker they can resolve some of those symptoms and the better recovery they might be able to have. Okay, now we're going to shift gears away from our brain and spinal cord, the CNS, and now we're going to talk about the PNS, or peripheral nervous system. Remember, the peripheral nervous system consisted of everything outside the brain and spinal cord, and there's two divisions here. We have a sensory division, which brings information into the brain, and a motor division, which brings information outside of the brain. That motor division is subdivided into a somatic division and an autonomic division. The somatic division deals with voluntary skeletal muscle contraction. The autonomic subdivision, on the other hand, involves involuntary body actions, for example, constriction or dilation of the pupils. Okay, while we're on the topic of the peripheral nervous system, I just want to say something about the difference between a nerve and a neuron, because this is a very common misconception among my students. So firstly, we already talked about a neuron. A neuron is a cell which is specialized to generate action potentials or nerve impulses. It has a cell body, it has axons, it has dendrites, and the important thing about this neuron is the cell bodies are always located within the central nervous system. A couple of exceptions, but for the most part, the cell bodies are either in the brain or the spinal cord. Now, the axons can travel well outside the CNS and oftentimes do. For example, this would be a motor neuron, and its axon is traveling to skeletal muscle fibers. So the muscles in your hands are actually innervated to neurons that have cell bodies way back in your spinal cord. Now, a nerve, on the other hand, is not a cell. A nerve is an organ. A nerve is a cord-like organ of the peripheral nervous system. It consists of bundles of myelinated and unmyelinated axons that are wrapped in these structures called fascicles. The important thing is these are just axons. There's no cell bodies. The cell bodies, where were they? They were in the CNS. So a nerve is a three-dimensional structure. It's large. You can see it with the naked eye. Neuron, not so much. But a nerve, you really can. There's some nerves in your body that are the size of your thumb. That would be probably the sciatic nerve. Now, most peripheral nerves are what we call mixed nerves. What that means is they contain both sensory axons and motor axons. So if we looked at this individual nerve, we'd see some information traveling this way, that would be sensory, some information traveling that way, that would be motor. So mixed nerves have both sensory and motor axons. Okay, now we're going to talk briefly about the process of sensation and perception, and this involves both the peripheral nervous system and also the central nervous system. So imagine that you have stuck your finger with a pen or something like that. Now when you do that, you are actually stimulating modified nerve cells out there that have free nerve endings within the skin itself. And this is where a process called transduction occurs. Transduction is where we basically translate a physical or chemical stimulus into a pattern of nerve impulses. So here's where we form our action potentials. Those action potentials will travel down our sensory neuron, and eventually that sensory neuron is going to connect with our central nervous system, here our spinal cord. And the spinal cord has other neurons, interneurons, which are going to help relay that information up to the cerebral cortex, and that's where perception occurs. Perception is identification of a stimulus and recognition that, oh, I just poked myself with a pen or something like that. Okay, before we end this lecture, we want to talk briefly about the autonomic nervous system. When you hear the word autonomic, it sounds like automatic. So this is our automatic nervous system. It involves sensory information that we're not consciously aware of, and it involves decisions that go on inside of our body that we also do not have voluntary control over. So this is controlling things like our heart rate, our respiratory rate, controlling our pupil constriction and dilation, and these are all automatic body functions. And this is out here in the motor division. So the autonomic division is subdivided into two antagonistic subdivisions, the sympathetic division and the parasympathetic division. Okay, our first subdivision of the autonomic nervous system is something called the parasympathetic division. The parasympathetic division predominates when we are relaxed. So when you hear parasympathetic, I want you to think rest and digest. These are things that are going on when your body is relaxed. And we divide these into SLUD responses. SLUD here stands for salivation, lacrimation, urination, digestion, and defecation. All of these processes are increased when we're very relaxed. Think about salivation. When you're relaxed, you have plenty of saliva. Lacrimation means tear production. You produce plenty of tears. Urination and defecation, they don't need to be explained. But think about how often you might urinate or defecate when you're at home on a nice relaxing weekend versus when you're out doing errands or at school or at work. You probably urinate more at home. On the other hand, parasympathetic nerve impulses can also decrease other things. For example, they help to decrease the heart rate. When you're nice and relaxed, your heart rate goes down. They also help to constrict airway diameter, and they also restrict pupil diameter. Your pupils become smaller. So now I want you to think about things that make you relaxed. What makes you relaxed? What do you do to unwind at the end of the day? Maybe you go out and you do some meditation. Maybe you do some yoga or something like that. So as you do, you breathe in and out, and in and out, and you become at peace. Your heart rate drops from 80 down to 60 or even 55. Your pupils begin to constrict. You think about just good things in the world. You breathe in, you breathe out, and then get you. Okay, maybe that's not Okay, our next subdivision of the autonomic nervous system is the sympathetic division. And the sympathetic division deals with E situations, that is emergencies, embarrassment, excitement, and even heavy exercise. So what happens during the sympathetic nerve response? Well, first of all, our pupils become dilated, and that allows us to bring a lot more light into the eye so we can really see what's going on. If there's a tiger chasing us, we can see the tiger from much further away. It also helps to elevate the heart rate and blood pressure. This helps to give us the energy we need to fuel our muscles to get us away from whatever the emergency is, the tiger, whatever. It also helps to provide us with a lot more blood glucose, so we have energy, again, to get away from whatever the emergency is, and it also dilates the airway diameter so that we can breathe much more easily and get a lot of oxygen to our tissues. Now, what does it decrease? Well, sympathetic nerve impulses decrease digestion, and they also divert blood flow away from non-essential organs. So if you're getting chased by a saber-toothed tiger, we really aren't concerned about how quickly you're digesting things. So we basically shut down blood flow to the stomach and so forth. And so as a result, if you're doing something that involves a lot of sympathetic nerve impulses, like even heavy exercise, it's very hard to digest things, and you can actually get indigestion pretty easily. So think about things that lead you to be excited or scared or something like that. Think about movies. Over here we have Shelley Duvall in The Shining, right? She's getting chased by Jack Nicholson, and he's like, "Here's Johnny!" As he's going through that door, the actress was actually traumatized. They did that scene so many times, and you can imagine that even if it's just an actor on the other side is coming in there with an axe and stuff like that, it's got to be a very, very scary situation. Take a look at this artwork. I think the Scream, you've seen this before. What does it look like? First of all, the mouth is open, the eyes are really wide, the pupils are dilated. This is a classic autonomic sympathetic nerve response. And let's take a look back at our guys back here with the bat, over on this side, right? That bat is hurtling towards them at breakneck speed. What are they doing? Well, they've got their hands up to protect themselves from the bat. What we can't see is most of them are wearing sunglasses, but definitely this guy, his eyes are fixed right on that bat, pupils are dilated, and their heart rate is racing. So think about the last time you were involved in an almost accident in an automobile or a bicycle or something like that. And if it scared you a lot, what did you feel like at the end of that situation? Now, even though you weren't hurt, you'll probably notice that you're breathing heavily. Your pupils are dilated. Your heart is just do-do-do-do-do-do in your chest. And the other thing is you're probably shaking a little bit. And that's because one of the things that happens as a result of a sympathetic nerve response is we secrete a lot of epinephrine and norepinephrine from our adrenal glands. So not only are these neurotransmitters, they are also very potent hormones that cause your heart rate to become elevated, that affect your respiration rate, that affect perspiration on your skin, and also just get you jacked up to deal with whatever the emergency is. So you might notice that this sort of situation perpetuates itself for the next 5 or 10 minutes or so until you're able to calm down. So big picture here is that parasympathetic responses deal with restful situations. They accentuate digestion, defecation, etc. On the other hand, sympathetic nerve responses deal with emergency situations or exercise. And they basically facilitate a rise in blood pressure, heart rate, all these things so we can deal with whatever that emergency is. All right, you've reached the end of the lecture on the nervous system. As always, if you have any questions about the lecture you don't understand, go back and review that part of the lecture again. If you still have questions, then send me an email or a text. Talk to you later.